you very much.
We can move now to the presentations. So I kindly ask the chair to announce the first speaker of the presentation.
Okay, we can start this domain. The first communication with the title Standard Operating Procedures for the Analysis of Microplastics in Larval Fish Diets. The author is Maione. Is there?
Yeah.
Okay, so the floor is yours.
Okay, thank you very much for being here today. Just very briefly, I'm a PhD candidate at the Polytechnic University of Milan and in industrial engineering, and at the Department of Geography at San Diego State University, and collaborate with the Metabolism of Cities Living Lab with my colleague, Dr. Domenico and Dr. Gabriella. And for today's presentation, I'm going to present a protocol for the analysis of microplastic in larval fish. So this research, as the contribution of this research is methodological. And so we started analyzing the open gaps and what are possible like objectives in the analysis of microplastics. So what are the open issues that we try to address, such as the limited data on ecological and biological impacts of plastic in native species, the uncertainty about like the translocation of microplastics across trophic levels, and some of the survey questions we try to answer were what are the abundance, distribution, and variability of microplastics? What is the incident of encounter between like microplastics and native species in waterways? And how does plastic affect subsistence and health in native species? And the objectives were to quantify the smaller fragments present in the digestive tracts of fish and to assess the possible specific impacts. So some of the acknowledged risks from the ingestion of microplastics are pertain to the biological effect with impact on the energy intake, respiration, and growth of the species, ecological effects, including translocation across trophic levels, survival, and chemical contamination, and also behavioral effect, which can change the feeding behavior of fishes. And the reason why we chose like larval fish for this analysis was to see if ingesting microplastic can determine changes in the feeding behavior when fish leaves the nursery environment. Okay, so in terms of like sampling methodology, there is mainly like water sampling and fish sampling, and in this case we apply fish sampling. And in literature, the main types of analysis for the analysis of microplastic can be morphological analysis, such as microscope analysis, which we use in this case, scanning, electron microscope analysis, image, and automated analysis. And in terms of chemical analysis, we can have spectroscopic analysis and thermal analysis, among others. For this standard operating procedure, we conduct a sample collection, we remove the fish diets, we prepare the slide, and we analyze the slide under the microscope, and then we use the software Image Pro to have like a computer-based image analysis and finally statistical analysis and modeling of the results. So I'm going to present the main stages of an application of the use of this like procedure. And in this case, we analyze like some native species of fish and trying to monitor the microplastic ingestion based on the classification of Free et al. 2014 that characterized microplastic in beads, which are present hard and rounded characteristics, fibers, which present themselves as like thin fibers and linear fragments. Fragments which are hard jagged pieces of plastic, films which are thin and filmy particles, and foam lightweight sponge-like fragments. For this, before preparing the sample, we prepare some like questions So like, how do you choose like the sample selection? So how many, for instance, some question that you can ask are how many samples you need, which samples type is relevant, should samples be collected within or across species to observe any like differences in ingestion patterns, and and in this case, like how do you interpret the results? Do samples' natural habitats differ? So what are some of the specific characteristics and exogenous factors that can influence like the ingestion? How does this affect the interpretation of the results? What are the external factors that can be considered when choosing the sample and can characterize your choices? And what are some of the limitations that you need to take into account? For what concerns the sample microscope analysis, which polymer types are accounted for, and how do you distinguish different types of polymer, which findings types, if you want to enumerate particles or like classified by particle color, size, weight, or visual coverage. What are how are the microplastics sorted from other like organic content in fish diets, and what is the best technological solution for the analysis to avoid biases and overcome the limitation that you found in the previous part? And also, what are some of the limitations instead that you need to take into account? And finally, for the sample statistical analysis and interpretation of results, some of the questions that we asked were what statistical tests are needed, which graph chart types are used to explain the findings, which type of findings are significant to the study, like also depending on the objectives and the specific goal of the study, which statistical errors are accounted for, and what are the limitations of the methodology used. So for this case, we analyzed 3 different sites. They all belong to the same region, but they have very like distinctive characteristics. So for instance, for site 1, it is a connecting channel, which is highly used for tourism purposes, and is also a site for commercial and recreational fishing. For site 2, we use a distributary channel connecting smaller lakes and wetland. This area is used for ecological restoration purposes and also for recreation purposes. For site 3, it is an urban river with highly present of anthropogenic influences, such as like municipal wastewater, sewage, runoff of chemicals and hazardous materials coming from the city. And also is like heavily polluted by industrial activities, fishing and shipping happening like near or like in the river. So the purpose for choosing these 3 different sites was to understand if being like so different in nature, but like all in the same region, we could notice any particular differences in the concentration of plastics in the 3 sites. For the species that we sampled, for site 1, we sampled 20 rainbow smells and 28 burbuts. For the second site, we had 30 yellow perches, 30 rainbow smells, and 28 burbuts. And for the third one, we have 20 cyprinate, 30 rainbow smells, and 33 yellow perches. And all of these are like native species for the sampling areas. So just very briefly to show you like the concentration, as you can see, we found microplastic to be abundant across all species and all sites. And you can see that like yellow perch has the highest percentage of ingestion rate, and also like in literature, like yellow perch is considered a fish, which is like very highly to ingest microplastic upon encounter. As you can see, the most present type of like microplastic was fiber in all cases, followed by fragments, and we only found evidence of a few beads. And in terms of colors, our color scale ranged across like 5 different colors, which were orange, black, blue, red, and white or transparent. And as mentioned, this slide is average size of the fiber, and the most dominant colors were blue, black, and red. So a possible reason for these colors can be found in literature, which says that light blue is also the color of like zooplanktons and other microplates, which is like which are like often found in the nursery environment where larval fish feed on. So this can like mistaking like microplastics for like microplates can be like one of the possible reasons behind these high ingestions. As mentioned before, we found the yellow perch reported the highest ingestion, but surprisingly, we did not observe any substantial differences among the 3 sites. So some of the consideration could be for future analysis to compare the incidence of like encounter with microplastics, with also some water sampling to understand like what is the percentage of like microplastics in water sample, to understand if fish like has to feed on plastic, or like if it's like a choice and a change in feeding behavior related to the like resemblance of microplastics and the micropraise. And often the methods can also like determine like our ability to understand and to trace microplastics. And also like this methodology offer like an understanding of like the types of like micropollutants that we can find in the digestive tracts, and this can have implication to understand like future changes since like when the fish leaves the nursery environment. Okay, thank you very much for your attention, and I'm open for any questions you might have.
Thank you, Carol. Any question?
Thank you for your presentation. I have 3 questions. The first is, do you think that method can be used for more microplastics than the ones which is the range of microplastics you can use the method for?
Yeah, well, in terms of like the accuracy of the methods, I think it also depends on like the type of microscope you use. For instance, for this analysis, I use a dissecting microscope with a 40x magnifying capacity, and then I couple it with the Image Pro software to further understand like the length, the area. But of course, like depending on the type of like equipment that you have, you can reach like even smaller size.
Thank you. And you extracted the microplastics from fish digestate, right? Do you think the method can be used for other substrates? I'm imagining something like soil or water.
That would be like really interesting. The purpose for this study is to use the digestive tract was to see if there is any changes in feeding behavior. So this was like the preparatory study and the methodology. But I think it would be really interesting to study other like sampling environments to see if the concentration of like microplastic is a determinant of like ingestion. So that would definitely be like an input for future studies.
And the last thing is, did you consider using something like Python for automatic data analysis with something like OpenCV to analyze the images? Do you think it's something possible?
Absolutely, that would be possible. I only briefly presented, we use like RStudio and SPSS for this analysis, but of course, because it was mainly like a methodological contribution, that would definitely be usul.
Last question, how did you catch the fish?
I didn't do the collection part myself. I only did the analysis.
Okay.
Yeah.
Thank you very much.
They were like really small fish. Like the longest was like 12 millimeters. So.
Even more difficult to catch.
Yeah. Any other questions? Okay. Thank you very much for your attention.